morning. Hi, everyone. On this nice rainy day, let's take a nice deep sigh of relief. We're here, we're dry, we're ready for systems biology. Today's topic is multi-objective optimality, or evolutionary trade-offs. And a subject which carries in it a lot of wisdom also for our own lives, not only for understanding molecular biology. All right, so we did the welcome and the breath. And I start by reviewing. It's as you remember in our course, after doing network motifs, then talking about robustness, we're now in a period of lectures about evolution and optimality. And the first lecture in that topic last time talked about the concept of the fitness function, where if we look at a trait of an organism, like for example the expression level of a protein, on the x-axis and the y-axis we plot fitness. We get a landscape. It could be a complicated function or a simple function. And natural selection tends to go to the maximum. So this is the trait we're measuring. Let's say the expression level of a protein. And here is fitness. And this is the fitness function, or fitness landscape, also sometimes called. In natural selection, if there's a mutation or change that makes the organism have more babies that survive, that mutation tends to increase in frequency inside the population, and fitness increases. So there's a force in natural selection that tends to take you to the optimum, x optimum. And last time we talked about an experiment to try to calculate the fitness function in a simple case, the lac operon, where x is the expression level of those proteins. Fitness there was contrived to be just the growth rate, an exponential growth rate. And we calculated the cost and the benefit of making the proteins. If the environment contained the sugar lactose, there's a benefit, but there's also a cost because the cell grows slower if they need to make proteins. And we saw that there's an optimum, and then we calculate that optimum and then evolve E. coli in test tubes day after day, diluting them again and again, and see how quickly they accumulated genetic changes that heritably brought them to this maximum. And we saw that in a few hundred generations, they came to the optimal level. And and they had many different solutions in the same test tube. So that was last time. Okay, so today we're going to make things more interesting by talking about what happens when fitness is made of several conflicting components, not only growth rate, but also something else. Multi-objective optimality. Before I do that, maybe I want to see if there's any burning questions from last time. I'm giving you plenty of time to think about a question as I'm erasing the blackboard. I'm going to demand a question, so one of you has to volunteer for the entire class and ask a question. Yeah.
In your experiment with the E. coli getting to the optimum, would they get there faster if they had a higher mutation rate?
Okay, the question is, we saw that E. coli generation after generation, their protein level changed. They reached the maximum. They grew faster. Would this happen faster if the mutation rate was faster? So, E. coli has mutation rate 10 to the minus 9 per base pair per generation, or one mutation per genome every 300 generations, this works out. And indeed, it's possible to increase the mutation rate of E. coli. For instance, there are E. coli mutants called mute that have higher mutation rates. These are mutations in the mechanism that repairs the DNA. The same kind of mutations are linked, for example, with breast cancer in human beings, because cancer also happens by mutations. And evolution, it does go faster when you have a mutator strains. So, for instance, an experiment took those mutator E. coli strains or bacterial strains and show that they can infect mice faster than the non-mutator strains. But what happens is they infect the mice faster, they grow there, and then there's a complementary mutation after they take over that makes them again have low mutation rates. That's a cool way, you can say, that evolution, natural selection, increases the mutation rate and then decreases it. Because you don't want a high mutation rate is also deleterious, which means bad. If you're here, then mutations are bound to take you down in fitness, no matter which way you change. So that's called mutational load. If you have a high mutational rate, the net effect of the loss in fitness because of mutation is called mutational load. So if you're in this region, mutation rate should be low. If you're in this region, high mutation rate helps you. And another secret I'll tell you is in those evolution experiments, and the first day, in the first test tube, there were already the mutants that were going to take over the population. There's so many bacteria there. They just had to take time to amplify like that. So, and the Erez Dekel, when he designed those experiments, he designed all the volumes of the serial dilution experiments to make sure that there's a chance that there'll be some mutants happening. So that's also like a trade-off situation. You don't want to make 100 liters every day because then you have to carry all that liquid. As an experimentalist, you don't want to do that, right? So it's 10 microliters. It's like it's one of those orange cap tubes that look like this. You have this much liquid in it. That's reasonable. Okay, but yeah.
So can you, or is there a way to quantitate the promiscuity of mutation rate with the distance from optimality?
The question is, can you correlate or calculate how much the mutation rate is and your distance from optimality? This is already, if you do that, I think you would be able to publish a paper. That's new because it's like a nice sigh of relief for the wet people coming in. Not wet. Is it raining yet? Still? Because it's very rare to be able to even evaluate this function. So, but why not? Okay, so thanks for the question. And now we're going to deal with trade-offs. Okay. Is this fitness function landscape? And it comes from a picture where we have the DNA, which biologists call the genotype. And that makes the shape of the animal, or the traits, which biologists call the phenotype. And that gives you fitness. For example, if you have this Darwin's finch here, you have the beak. The DNA gives rise to the developmental process. It gives you the beak shape. The beak eats these seeds. The bird has food, it can make babies, and that gives you fitness. So if you plot, if you try measuring the beak, measuring different traits from the beak, these are things you can measure. You have what's called the shape space of the beaks, let's say, or more scientifically, morphospace. Morph is shape. And inside the shape, I can plot this fitness function. I'm going to plot as a topographical map. So those of you who can read topographical map recognize this is a hill or a valley, right, by telling you it's a hill. Okay, so this is the maximum. And this is the fitness landscape. Natural selection will go here to the maximum, let's say, or attempt to. And that's the beak that we'll see. But I want to ask you, if the beak, or whatever it is we're studying, needs to perform more than one task, no shape can be optimal at both tasks. For example, suppose in this lifestyle of this bird, needs to eat seeds, but also needs to make a living off pollen or nectar or insects from a flower. So you can think that the optimal beak for seeds maybe looks like a plier. But the optimal beak for flowers maybe looks like a nose plier, right? And a single shape can't be optimal for both. And that means we have a fundamental trade-off if you have more than one task. So we need to modify this picture a little bit. And what I'll talk about in this class is how can we understand trade-offs like this. Nice deep sigh of relief. How can we understand it and show you that it leads to special, actually to special kind of simplicity in what natural selection, in the range of shapes found in nature.
Is there some sort of shape that is good enough for both tastes? Can you define it good enough for both tastes?
So the question is, we need to find a shape that's good enough for both, or a compromise, and that's what we'll discuss. Sometimes yes, sometimes no. So that's the question, and so we're going to modify our basic view by talking about DNA that makes the phenotype, the shape, and this the shape has performance at task 1, performance at task 2, and these combine together to give you fitness. Right? So we need to add this concept of performance functions between the phenotype and the fitness, or to break up fitness into tasks. And in general, you would say that now if I have my morphospace here, and I measure things about the beaks, every point here is a possible beak, right? Could be that there's some constraints here, like, the beak. If this is one of the traits, this is the mechanical strength of the beak. We can't have a beak made of titanium yet, right? In the Darwin Finches, because it would take them much longer to evolve that. So there are constraints built in here. What we can do is now in the lab, in principle, we can take beaks, make the beaks out of some material, and test their performance at crushing seeds, let's say, and picking insects. We can do that, let's imagine, or proxy for that, and look at the performance function. So I'm going to draw here, for example, this is the hill, the performance contours for task 1. Let's say eating seeds. And here is the performance contours for task 2. Let's say eating pollen. So we have these performance functions. We need to one performance function for each task, and we're going to call the peaks of these performance functions the archetype, which is the best phenotype for task 1. And This is the archetype for task 2. So these maximum of the performance functions, we call them archetypes. And just mathematically, fitness is an increasing function of the performances. So fitness is some increasing function of the performances. This is the performance function P1, this is the performance function P2, and fitness is the increasing function of performances. You can say it weighs or combines the tasks according to the specific niche that this bird is living in. So if you're eating seeds, the fitness function gives more weight to this performance function. And if it's eating insects, it gives more weight to task 2. And in a situation where you eat both, it's some combination of those two performances. So if we look at a certain niche, the fitness function combines these two, and you get a kind of... So let's say in a niche where seeds are important, you combine these two, and you might get some kind of contours like this for the fitness function. And you notice that the maximum is kind of close to the archetype 1. And if I had a niche where pollen is more important, the fitness function, which is a combination of these two, will be shifted up to here. And if it's a generalist that needs to do both, fitness function will have a maximum somewhere in the middle. Did I explain myself? Right, so we have these performance. We need to worry about the performance functions. By the way, it's much easier to measure performance functions than fitness, because fitness you have to go to natural world and observe the babies and their survival, etc., over generations. Performance functions, in principle, you can measure in the lab if you have an idea what the tasks are.
Do they always have to be on the same line?
Okay, so that's the question is, I drew it here like on this line here, and that's where I'm going. That under some conditions, it's on the line that connects the two archetypes. Yeah.
How does this fitness function relates to the earlier defined fitness function for the E. coli, or just the expansion rate, or
So I'd like, yeah, question, how do I connect this fitness function to what we talked last time, where I said fitness there, I'm forcing it because of the experiment to be just one task, which is exponential growth. So there I'm forcing fitness to depend on one task, which is exponential growth rate in test tube. But you can imagine that E. coli has another task, for example, to survive against stresses, et cetera, et cetera. So in the real world, exponential growth rate is not the only component. So what I did in the last lecture, actually, was work in the regular paradigm of just a fitness function, which in fact means I'm just considering one performance function, which is exponential growth, and saying that fitness is proportional to that. And that's the way most papers until recently dealt with evolution. But if you want to add an additional task, you need to do multi-objective optimality. I see I didn't answer your question.
maybe I need to ask a question again. Last time you were talking about some specific protein expression that was depending on a specific gene. Here you're talking about the shape of the beak that apparently has this genotype-phenotype relation, but it's an ensemble of many genes and the expression of a lot of things.
I see that, okay.
The mutation.
Okay, now I understand your question a little better. You're asking, last time I talked about such a simple system, just one operon, and the phenotype was eating sugar. Here I'm talking about something completely different, the shape of a beak, which depends on many genes. And how does evolution work in this situation? Is that what you're asking?
Should scale with something, I don't know with what, but it should be very, very slow because you need to have a lot of genes that each
Oh, I
one
see.
need to have its own mutation and a
Okay.
specific.
So you expect, because there's more genes affecting a beak, let's take a nice deep sigh of relief, then this E. coli gene, maybe evolution will be slow.
Or it will not happen at all.
Maybe it won't happen at all. Yeah. So that's good. And that's indeed, before there was data, there were a lot of controversy about how slow evolution can be in a multi-complex, multi-gene trait, like shapes like this. In fact, that's what Peter Grant and Rosemary Grant, when they studied Darwin Finches, and they sat on the Galapagos Island that's about as big as from here to the Wolfson building. They lived there for decades. And they counted every bird there and measured their traits, etc. They could observe natural selection in the beak shape and working on the scale of years, which means a few generations. There was a drought, only big hard seeds remain, and the gene frequency shifted so that the birds with the big beaks were much more frequent. Then there was a year with a lot of rain, and the birds with the big beaks declined, and the birds with a small beak. So they could show that natural selection works there on the scale of a few years, a few generations. And you can say, how is that? And the reason is, it doesn't have to wait for mutations. What it does is recombinations. You take the parents. They have a lot of differences, the parents. Each, like I said before, each of us is a unique experiment. Our DNA has never been tried before. And by combining parents, you can increase quite quickly. And the selection is very, very strong there, because you can have 1,000 birds of a certain species, and then there's a drought, and it goes down to a few tens. So it's like a terrible bottleneck. Natural selection, if you have a small edge, you have a huge effect on your survival. And so because of this recombination, what's called of different parents, evolution on a multi-gene trait like this works very quickly. We can discuss that. I want to talk about things like that in this lecture too. And it's as if that system is designed to quickly change in certain directions, useful directions. And we'll talk about how that can be. So it's not only the E. coli that shows this rapid evolution. E. coli doesn't recombine there. It's even, I would say E. coli is slower than the rate of change of these things. It's not hundreds of generations, it's just a few generations. Because E. coli doesn't recombine, it has to rely on mutations typically. Even though in the natural world E. coli can take DNA from other bacteria, we have another opportunity for a nice deep sigh of relief. Our lungs fill up with oxygen. Yeah.
Could it be that there is no optimal solution? Like if you're trying to weight both performance functions, you get to a point where you're not good enough in both?
So the question is, maybe there's no optimal solution. You're not good enough in both. Well, let's discuss it, okay? I mean, it could be, if these performance functions are extremely steep like this, then the points in the middle here are not viable. And then you'll have either this or this.
But then you lose one of the performance.
Yeah. I want to discuss cases where things in the middle can kind of do a little bit of both. I mean, okay. So what happens in this situation? So as already somebody says, you expect the best compromise to be between the archetypes. So I want to tell you about a theorem. The theorem is from my group, Oren Shoval, Science 2012. You can read that paper, and I'll tell you a lot of things from that paper and beyond. So here's the theorem. It says that if it has conditions, right? If the performance functions decreases with distance from the archetype, so this space has a distance, so you can think every point here, you can measure the distance of this point from this point. I'm talking here for professionals about metric distance, inner product metric norm. Then no matter what the shape, what the fitness function, right? As long as it's an increasing function of performances, the optimal solution is on the line segment connecting the archetypes. So I'm going to tell you what I mean right now. I'm going to erase this, okay? It's much easier than than you might. These are the traits. Could be many traits, multi-dimensional space. And here's the archetype of task 1. Here's the archetype of task 2. And let's take a point here. Some point I choose arbitrarily from morphospace, yeah, to write bigger. Right. This point A here, okay? This distance determines basically its performance at task I, task 1. The farther away it is from this archetype, the worse it performs in task 1, because this archetype is the best. This distance determines its performance at task 2. If I draw the line between the archetypes and draw this point here, this point B, you see by geometry, it's closer to both archetypes. And therefore has higher performance in both tasks. And therefore it has higher fitness, because fitness is an increasing function of performance. So if you're better at both tasks, you obviously have higher fitness, no matter what your niche. And therefore, we can erase point A because natural selection will prefer point B. No, if I make a competition between them, no matter what the niche, B is better at both tasks. Now, since there was nothing special about point A, right? So we can erase actually all the points in morphospace except for the line segment between the archetypes. So what we can do is erase all. I can do this for every point. And what I'm left with, the only points I can't erase are the points on the line. And this proves that the optimal solution, no matter what the niche is, has to be on the line connecting these two archetypes.
It's not necessarily a straight line. The form function of the radial symmetry.
Okay, so what I told you here is a theorem. Always theorems have some conditions if performance decreases with distance. So that means the contours of the performance functions here have to be like circles or ellipses. Yeah, if there are ellipses which are with the same distance metric, so the same ellipses, it's also a straight line. Also, if there are ellipses in 90 degrees, it's a straight line. And if there are general fitness functions, performance functions of general shaped contours, the line can be slightly curved. So relaxing the assumption of the theory can lead to slightly curved lines. And here's where Hila Sheftel, who's sitting here, joined this effort, and she could prove that if it's two general ellipses, this line looks like a parabola segment, but it's not very far from a line. And it's hard to get far from a line if you try different performance functions. You can, but a line is a very good approximation. Okay, so just to make it simple, if you have two best designs, which are quite complicated, all the biology, all the complexity of biology is in the archetypes, which can have amazing design. But if you look at all the possible bird beaks, the simple part of it is they'll fall in a line. Just weighted averages, ממוצע משוקלל of the two archetypes. That's the simple part of the problem, the way things combine. And your position on the line is called Pareto Front. That's the technical name for it. This kind of multi-objective optimality where what you do is you compare 2 points, and if one point is better at both objectives than the other, you erase the point that's dominated. And you do that again and again and again, and you're left with only points that you can't improve in both tasks. That's called the Pareto Front. This is how you deal with multi-objective optimality. So this is called the Pareto Front, which means that notice that the specialists are at the ends of the line, specialists in one task, and generalists that can do both tasks in the middle. Now let's make it more interesting. What if there's three tasks? Suppose this beak has to eat large seeds, small seeds, and flowers. Or something. Okay.
You just get the volume that's encased by the intersecting lines.
So the answer is, if there's three tasks, first of all, if there's three tasks, I have three archetypes, right? Three performance functions, three archetypes. And the answer, with a similar kind of theorem, is that the optimum has to fall somewhere inside the triangle. So that's the Pareto Front in this case. And again, weighted averages of three archetypes make a triangle.
So then there's no optimal point.
So the question is there an optimal point? So I just want to stress again.
What are the axes here?
Traits. So for instance, big width, big height, big depth, this angle, this angle, this angle,
Okay, so this is in 3 dimensions. We were kind of
I drew here 2 dimensions. But if you have more traits, I'll talk about it in a second if you have more traits. And I wanted just to stress this point of optimality. It's very, very important that you understand. Performance functions, the maximum of each performance function is one of the archetypes. Now, if I have a certain niche, the fitness function is a combination of those performances. So if I have just one task in my niche, fitness is basically based on one performance function. And its maximum would be here, and I have a specialist that's here. But if I need to do all three tasks, fitness says, okay, it's 80% task 1, 20% task 2, and 10% task 3. I'll be somewhere here. So it is the optimum. The fitness function of that niche has an optimum. It's like a hill, has an optimum here. I now have another niche, it has an optimum here. I have another niche, I have an optimum here. The theorem says that I take all possible niches, all possible ways to combine the performance functions. I guarantee you that their maximum, their optimum, will lie somewhere inside this triangle. So it's a statement about all possible optima of all possible fitness functions based on those performances. Or if you look at all the birds in a certain taxonomic group and you record their beaks, each one is a point in morphospace, you should see these kind of shapes. And you can infer the tasks from the data. So this you don't have to know the tasks in advance by looking for these kind of polygons like triangles, lines, tetrahedrons, and seeing what is special about the animals or systems near the vertices, kodkodim. So if you find birds here that eat flowers, birds here that eat seeds, and birds here that eat worms or something, you can infer what the task might be. There's something you expect to see something special near the vertices. This is a way to infer tasks. Yeah.
So we say that the number of vertices is equal to the dimensionality of the trait space?
Is the number of vertices equal to the dimensionality of the trait space? No, it's equal to the number of tasks. Let me now go, because several people asked, what happens if the trait space is high dimensional? Let's just say I now measure 100 numbers about the beak. Okay, so if I measure many traits, it's called high dimensional trait space. So of course I can only draw 3 dimensions. So let's imagine I measured 3 numbers about the beak. Trait, the height, the width, the curvature. Suppose I have only 2 tasks. So in this 3-dimensional space, this is the archetype for task 1. Archetype 1. This is archetype 2. And what I expect to see is that the data will fall on a line between these 2 tasks. So even though I have 3-dimensional or 100-dimensional trait space, doesn't matter, the data should fall, if there's only 2 tasks, on a very low-dimensional object, just a line. Each archetype is a point in trait space. It's characterized by all those different traits. The other archetype is characterized by all those different traits. But the optimal solutions are guaranteed to lie on the weighted average of those 2 points on a line, or a slightly curved line between them. So 2 tasks means the data should fall on a low-dimensional basically polygon, or mathematicians would call it polytope, which is the generalization of polygon. It could be, if it's 2 tasks with number of vertices, or kodkodim, equal to the number of tasks. So let me ask you this. Suppose I measure many traits and I have 3 tasks. 3 tasks. What would happen to the data? Should fill a cloud in this high-dimensional space? It should fall in a triangle. Now, how many dimensions does a triangle have? 2. Triangle, you have these 3 points in this space. 3 points define a plane. And on that plane, they define a triangle. So the data should fall on 2 dimensions, which is, again, if you're measuring 100 traits, to have them fall on 2 dimensions is a remarkable coincidence, right? It's a big simplification. So if I plot these 3 archetypes here, These three archetypes define a plane, because any 3 points define a plane, unless they're on a line, which is highly unlikely, right? And on that plane, they define this triangle. So the data should collapse on a triangle. 3 tasks. Data falls on 2 dimensions. And if you project on those 2 dimensions, on a triangle. What happens if I have 4 tasks?
But is that okay for how many tasks, for how many dimensions? You're outlining 4 tasks.
Slow down. Slow down. If I have 4 tasks. 4 tasks, 4 archetypes. 4 archetypes define 3 dimensions. If I had 100 dimensions, this is again for people now is not used to high dimensions. It can start becoming make you dizzy. But it's also so you have a tetrahedron. Okay. What happens if you have a tetrahedron? The reality is 4 tasks. But your experiment only allows you to measure 2 traits. Okay, you can only measure 2 traits. So I can only measure, for example, so if I measured many traits, I would see a tetrahedron. But in fact, I can only measure these 2 traits. Like, I don't know what this one is. How would my data look like? It's like projecting the tetrahedron down to a plane. And when you project the tetrahedron down to a plane, it looks like a triangle. If this point happens to be projected here. Or if the tetrahedron is a little like this, it could look like something like this. So if you don't have enough dimensions, if you haven't measured enough dimensions, you still get these polygons. Okay? But sometimes sometimes it's difficult to tell if it's 3 tasks or 4 tasks. So you need if you have 4 tasks, you need at least 3 dimensions to get the full geometry. If you have 2 tasks, it's enough to get, you need 1 less dimension than the number of tasks you might have. So a triangle, it's enough to measure 2 tasks. So that's what happens when you don't have enough measurements. Yeah.
You assume here that the shape landscape is continuous, right? So there exists a point B. So you can erase A only because point B exists, right? Like there are a combination of trait 1 and trait 2 that
Okay.
I mean there are
All
some
right.
physical.
Good. Thanks. Okay. So the question is, or I guess what you're driving at is, could be a situation where point B is impossible. Yeah, and maybe we'll see a case like that. And so the theorem still is okay. It says, all it says is that no matter what the niche is, the optimal solution will be on the line. But it doesn't guarantee that you'll see the entire line. The line can have holes in it, or it can be populated. It's just it's still this point is on the line, but it's unfeasible. So if I have some constraints, let's say they chop up this space, let's say all this region is impossible, then only this part of the line that's outside the region is feasible. Yeah.
But maybe then you can see points that are out of the line because.
You can't see them.
What?
Right.
You can see them.
You can because?
Yeah, because there aren't points that are closest to the archetype
Okay.
and are feasible. So.
I got what he said. I think you're right. It's a good point. So suppose this is the Pareto Front, but for some reason there's a constraint, you can't be here. And your niche is such that if this was possible, this would be the selected point. But it's not possible, so it could be that evolution goes to this point, which is the closest, you can say, to the line. And then your Pareto Front would look like something like this, maybe. Go around. Maybe.
Possible, right?
It's possible, yeah. Thank you. So in fact, this is, I've been thinking about this for years, and I didn't think about this particular situation, so I thank you for this question. It's really, sure. I think that's what would happen in this case. So this is like, this would be some kind of physical constraint. I know of examples where we see the triangle, I'll tell you soon, and it's not populated entirely, there's like a region that's not populated. But that might be a situation like that. But where I have to think about that. Maybe you can think of some example. Do you have an example in mind? No? Yeah.
One possibility would be looking at dominant and recessive genes without polygenic traits.
Okay.
Yeah. So it's either you have dominant or you have recessive. In the middle, it's just not dominant. So I'm not sure if
Yeah.
anyone.
Right. You can imagine tasks where there's no way to be in the middle. Yeah. So that's if performance functions, you can say, are very, very steep. And then the thing in the middle will never be optimal, and you only get population of parts of the line. And I want to say that that's okay from this theorem point of view. It doesn't tell you that the entire line has to be populated. That has to do with things like compatibility of the different traits. Okay. All right, so we talked about basically, let's see where we are. We talked about this concept of the Pareto Front, that depending on the number of tasks, you get shapes with the number of vertices equal to the number of tasks. The vertices are the archetypes. The vertices are special. They're the specialists. If there was only one task in the world, that's the phenotype you would see. In the middle, there's generalists. They're also optimal. They're optimal in a niche that needs all the tasks. Okay, so that we're not discriminating against those in the middle. We're saying they're also optimal. It just depends on the task, right? So those are generalists. They're not optimal at any task, but they're optimal at multitasking in that particular niche. And then there are generalists, specialists, which are optimal at one task, which are the edges. If you look at this shape, you should be able to infer the tasks by seeing what's special about the animal or systems near the vertices. Okay. If you don't see something special near the vertices, maybe you see a line or a triangle for a different reason. Could be, right? And we talked about this is one explanation for why we see low dimensionality or can't see low dimensionality in high dimensional data, which is a blessing for human beings because, as you can see, we can't picture anything more than 3 dimensions. So if our data is 15 dimensional, we can't use our vision and all our metaphors like that. So it's nice if data is low dimensional. It's simpler to understand. This is a reason for it. And we talked that the lines and triangles could be slightly curved. And now I want to give you some examples from real examples, and we can discuss what happens to them. But you know, I'm thinking maybe it's good actually at this point to take a break, a 10-minute break. So please come back at 10:15. Let's take a nice deep sigh of relief, and we come back at 10:15. We learned in the break. Let's start again. Let's take a nice deep sigh of relief. So, what's your name? Howard. Howard said this is the Pareto Front. Parrot is a Tuki. There was for those of you who are interested in math, you can say that the theorem is that the Pareto Front is the convex hull of the archetypes. Convex hull is a way of saying, so if I have these points in space, and I take a big elastic sheet, and I let it shrink until it can't do it anymore, that's called the convex hull. So if I have 3 points in space, that sheet is going to end up in a triangle. If I have 4 points, it's a tetrahedron. So it's always convex. Even if one of the archetypes is in the middle, the Pareto Front will look at the most convex shape it can make out of the archetypes. And I want to now give you some examples for morphology and gene expression, and then I think some of your questions might be addressed. And I sense some of you are quite excited, and so am I. I'm so happy with this Pareto Front, I have to tell you, because all right. Oh, another thing I learned. in order to do this analysis, you don't need to know what the tasks are or assume what the dimensions are. No. You take your data, measure as much as you can, ask yourself, is it a cloud, or does it fall on a line or 2 dimensions or 3? And if it falls on let's say on a plane, you say, aha, maybe this is relevant. If I look on that plane, does it fall in a triangle? Yeah, it does. Look, there's kind of a triangle here. Is there something special about the things near the vertices? Yes. Maybe this theory is relevant. So it's a way to discover things from the data. You don't need to assume that you know the task, for example, or the dimension. It's kind of a bottom-up. Yeah.
Question: is it always like flat surfaces, or is it sometimes like when you do like soap bubbles and they like?
So is it always flat surfaces, or can it be like this kind of thing?
Like
Yeah.
when it takes the lowest form.
I'll just say again that this theorem talks about a convex shape, so you can think about those kind of shapes and ask if are they convex. And look, so some examples. So I'm going to tell you some examples from the field of morphology, which is measuring animal shapes, and we're going to talk about the 3 molar teeth in rodents. Kathy Kavanaugh, by the way, is a co-author on this paper, and she's a zoologist, who showed me graphs like this that got me interested in this problem. So field of morphology is a beautiful field with a lot of mathematical analysis. And you have 3 molar teeth. You also have 3 molar teeth, you know? M1, M2, M3. M1 is right back here. And morphologists, what they do is they measure things like the area of the shape of the teeth, and they work in dimensionless combinations to normalize out the size of the animal. So here we will plot, let's say, M2 over M1, and here's M3 over M1. These are the tooth area. All right. And each point here is a different species of rodent, Mechalsem. And it looks like this. So first thing you see is that most of morphospace is empty. Data falls on the line. And that's well known in morphology. Most of morphospace is usually empty, even though you can certainly imagine a tooth configuration here. Why not? But it doesn't occur. And you get these lines, and sometimes they're called allometric lines if its mass is one of the coordinates. And lines are the way you take the data and you fit it to a line in morphology a lot of times. And now I want to tell you, if you look at this data, you see that these rodents eat animals. These rodents eat plants. And that these ones here eat both. So the rodents are arranged on this line according to their diet. The teeth for the plant eaters, the archetype for the herbivores is equal-sized teeth. For the carnivore, it looks like this is the archetype. This is extrapolating to this non-existent point where you lost your entire third molar, actually. And this line here, has the property that always molar 2 is molar 1 plus molar 3 divided by 2. That's the property of this line. You can see it from these 2 points here. So if I take, and this actually also applies for dinosaurs. So if you find the middle molar of a dinosaur, you can tell the size of the animal, basically. So that's a rule in paleontology. The middle molar is the average of the two. If you find a tooth, you can basically tell what the animal ate because there's also a lot of morphological correlates. These are flat for the plant eaters and they're pointy for the animals. But this is just to tell you an example of a 2-task Pareto Front, right? You have the herbivore, the carnivore archetypes, and you have weighted averages in the middle. But an open question for me is, if you take the entire topology of the tooth, and there's databases with 3D pictures of these teeth, is the entire weird topology also a weighted average? I'd love to see that. That's a project to be done. The data exists. Okay, so I just want to say, this is a line, but if I plotted here this squared versus this, it would turn into parabola. So what happens with the theorem? Straight line. So the theorem talks about you need to work a little bit as a morphologist to find the coordinates where you get a line. Those are the coordinates where the performance functions kind of look like how they have a metric. And indeed, this takes some knowledge of the system. And morphologists have that knowledge. They know which coordinates give you lines because they're looking for lines. But there was not really an explanation for the lines. So you have to know, you have to work on your coordinate system and know your biology. What's the most robust measurement, etc. Okay. And there's plenty of lines. Like the textbook's full of lines. And okay. Now let's talk about triangles. Can I erase?
Why does it always end to equal some 1% free hybrid?
Why is this rule? In my point of view is why this rule is because there's something about those archetypes. The archetypes, because of biomechanics, the equal-sized teeth, for some reason, are good for plants, maybe because they give you a good plane surface for crushing. And something about this 1 to 2 to 0 is optimal for ripping up the animals. I don't know why. But once you have those two archetypes, and I tell you it's weighted averages, it has to be that rule. Because that's only a line through 2 points. If both points have this property, then the entire line will. So that's my explanation, is the Pareto Front explains it. But if you, based on the biomechanics, the complicated biomechanics of teeth for the two archetypes. Okay, so Darwin Finches. So on this island in the Galapagos, Peter and Rosemary Grant, if you want to read The Beak of the Finch, I don't know how many of you have read this very interesting book about their life. It's a popular book about evolution. And they measured for each bird 5 traits for each bird over years. So it's a 5-dimensional morphospace. They measured mass, beak shape, leg length, things like that. And they observed this 5-dimensional morphospace. Each bird is a point here. The data falls on 2 dimensions. On a plane. And there's, for example, I'm just mentioning principal component analysis is a mathematical technique that can take data like that and ask, does it fall on a plane, on a triangle? I'm not going to get into it right now, but those of you that are interested should Wikipedia principal component analysis. And when you look at this plane, you basically see that the plane, you can describe it with one axis is the mass of the bird, and the other one is beak shape, coordinate. And you see the 6 different species, and the 6 different species fall on a triangle. These ones eat the large seeds. These one eats the small seeds. These one make a living off the flowers. In the middle are generalists species. Right? So here we have a high-dimensional data set, collapses on a low-dimensional data set. On that low-dimensional data set, so far, this collapse, you can look with principal component analysis. Now you add something to it. You look on that plane, and you see a triangle. You look at the triangle, and you look what's special about the birds at the vertices, and then you can infer tasks. And of course, there's a lot of data on bird beaks. The ones here have these big beaks. I try to draw one of them. These have this small beaks, looks like a small plier. These have these nose pliers. These in the middle have something in the middle. So there's different phenomenon here. Different species tend not to overlap. There's something called character displacement. So if two different species come to an island, the individuals kind of move away from each other. There's all these kind of cool evolutionary things you see when you... By the way, a single beak species colonized Galapagos Islands on the order of 1 million years ago and diverged into these species. And in different islands, different founder species diverge into almost identical 6, 5, or 6 species. So there's evolution replays itself when, and especially when you look when an island gets colonized. A different, let's say, lizard. There's a classic study in lizards, Losis et al. In the Caribbean. In each Caribbean island, Jamaica, Dominican Republic. I'm talking about reggae. A different lizard came there, different species of lizard. But on each of those islands, now you look, you see almost identical 5 species. There's this huge lizard lives on treetops, and a tiny lizard that lives on the edge of branches, and 2 species that live on the grass, etc. These same species were reinvented and each time a lizard came to one of those Caribbean islands, converging on almost the same forms. But you can tell that historically they came from different founder species. I love that. So it's like the tasks are physics. They're not moving. The task of living on a treetop or on the grass, that's the same in all the islands, more or less. And when the founder species comes, it has enough genetic variation to fill out the triangle. Again and again.
Maybe if you would measure more traits, the data would fall on a higher dimensional space?
If you measure more traits, data could fall in a higher dimensional space. For example, it could be that there are traits that have to do with tasks that don't have trade-offs. For example, the beak, there's tasks that trade off, like what you eat. But maybe the color of your feathers has other tasks. And those 2 tasks don't trade off. So you might expect that some traits would fall separately on a Pareto Front that has to do with camouflage, and other traits will fall separately on a Pareto Front that has to do with eating. And each bird is an outer product of your coordinates from the camouflage and from the feeding, and you can make combined birds like that. So it could be. And I expect that there'll be different Pareto Fronts for different parts of the animal. And that's something that we're still trying to understand. Darwin Finches. Okay, let's talk about ants. So this is E.O. Wilson, who invented words like biodiversity and worked on conservation. Still alive. And he looked at the leafcutter ants, and he plotted here something like their size and the relative size of the trail pheromone gland. So you have, this trail pheromone gland. Where are the legs in the ant? Do they come out of this segment? Who knows? Here? Like this? Makes sense? All right. And here, each point is an individual ant in the nest, okay? Each point here is an individual ant from the same nest, okay? And of course, once you start looking for triangles, you see triangles. But there's also statistical tests you can do to ask yourself, is the data more triangular than at random? And this, again, area of active research for us. But and Wilson also measured what these ants do. So these ants here, they are in the nest nursing. These ants here are foraging. You see they have a big relative size of the pheromone gland. And these ones here are the soldiers. Okay. Now I want to ask you guys, why are there ants in the middle here?
In case the rest die, so they can turn into the, so they can specialize if everyone else dies.
It's easier for these guys to do.
These ants in the middle are suboptimal, according to our theory, right?
But you never know which one you look at the process, the evolutionary process.
They do multiple tasks.
They're multitaskers. So the idea that Wilson has is that if you rigidly define, you want, let's say, 20% soldiers. If you had a cluster here, and a cluster here, right? Nothing in the middle.
Can it be a projection of another task that you have to affect?
Could be. So this is another situation you might expect, right? It's another situation. You have optimal soldiers, optimal forces. The problem is, like you said, if there's an attack, let's say, on the nest, and it's not enough soldiers, you need to make new soldiers. But it takes a while to make new soldiers. So according to Wilson, it's good to have a pool of generalists that you can be, gives you flexibility. Okay? And I just wanted to mention, this is also a metaphor for what happens in a multicellular organism. So we have different cell types, different tasks. And you can ask yourself, are there situations where you want the traits of the cells to have very rigid, like well-defined cell types, and are there situations where you actually want flexibility in terms of generalists, like less of a defined cell types or something. So this is, we're using this for
And these are clonal, right?
They're not exactly clonal. They're all the children of the queen. They're 2/3 genetically identical to each other, unlike me and my sister, which are only half genetically identical because of their genetic system. So they're kind of similar. But the selection bottleneck is on the queen. So this is an interesting kind of selection problem. Just to tell you a little bit about the difference between this communal selection and the kind of selection we used to for in bacteria. What happens if there's a mutation in the genome that makes now the soldiers, better at this task? What do you expect would happen to the fraction of soldiers in the nest over time? So if this was just bacteria and they're better at a task, those mutants will increase. But in the ant situation, if you're better at a task, that fraction will decrease. Because selection is the level of the whole factory, you can make less soldiers, get the protection, defense you want, and use those resources for the other tasks. So it's like a different, almost counterintuitive effect of when you improve in a task, depending on the level where you're selecting, we can say. In other words, cancer cells in our body are doing selfish evolution like bacteria. They're growing in number. But that's not necessarily beneficial it's not beneficial for the organism, right? That's if you had an animal whose red blood cells are more effective, it could reduce the number of red blood cells, still get the same amount of oxygen, and use those resources for something else. I hope I explained myself with this little Wilsonian, Wilson and Oster kind of arguments. Okay, so I think I'm going to skip the ammonites and go to gene expression. Let this simmer, percolate through your mind here. These morphological examples. And maybe I just mentioned this situation, if you have a high-dimensional data set, this situation is called clustering. And there's different clustering algorithms that can give you very well separate into clusters. But if this is the way the data looks, clustering algorithms that look for clusters won't work very well. They'll make some kind of arbitrary partition. And you need other kind of algorithms, basically, that look for these kind of polygons in order to understand your data. Saying this to those of you here who are dealing with high-dimensional data. How many here are dealing with high-dimensional data, like DNA microarrays, genes, love numbers? So say about a third, more than a third. The rest of you are dealing with low-dimensional data or no data? Zero-dimensional data. Okay. So So try to see if your data is low dimensional. And yeah.
Is there some sort of relationship between all the different triangles? Is there some sort of connection between all the triangles?
Is there a connection between the different triangles?
Yeah, all this data that's been analyzed. Something about area, perimeter, triangles.
Oh. I don't know of any connection. Okay, so I want to turn now to gene expression in cells. So here you might say, so the trait space I'm going to ask is how much protein does the cell choose to make from each gene? How much to activate each of its genes? Now you might say, this has nothing to do with the bird beak problem, because the bird beak, you're born with a bird beak and you can't change it. But gene expression you can change, right? We talked about transcription networks. You have a different situation, you can change your gene expression. You have lactose, you turn on the lac system. You have no lactose, you turn on the... So it's much more flexible. But there's still a fundamental trade-off. Because imagine that in a given second, let's say, in the next second, you can make, let's say, 1,000 proteins, okay? You need to choose which ones to make. Because you can't make a portfolio of proteins, a collection of proteins, that's optimal for, let's say, both growing fast and surviving stresses. For example.
Why not prioritize the tasks?
Exactly, you need to prioritize the tasks. You need to solve a resource allocation problem. You have the same kind of trade-off, fundamentally. So why not analyze it using the same kind of theory? Okay, so here in this paper you'll find an experiment like that, where we looked at E. coli, and we had a way of measuring all the transcription rates of all its 2,000 operons. Let's call it promoters in real time. And that's made by fusing the promoter to green fluorescent protein. And looking at 2,000 different E. coli, each one reporting, turning green in proportional to the production rate of a different protein. So here, on this axis, for example, is a promoter 1 divided by sum of all the promoter activities. So this is a fraction devoting to one of those 2,000 promoters. And here's for promoter 2, promoter 3. It's a 2,000 dimensional space. Okay, so we're not talking about low dimensions here. We're talking about serious high dimensions, right? With the curse of dimensionality, all the problems of high dimensionality. So again, very deep problems now in biology, how to handle high dimensional data. Because if you have 100 dimensions with information and 2,000 dimensions with random noise, you can imagine the difficulty of picking out the information from the noise, et cetera, et cetera. So it's really, really a problem. And so that's our morphospace now. Morphospace, each point here is a choice E. coli makes how to distribute its transcriptional resources between the different promoters. So we can ask, is most of morphospace empty? Or maybe if you look across different conditions, is it like a cloud? Does E. coli fill out the entire space? Or is it a low-dimensional situation? Are there polygons? All these questions, right? And if there are polygons, maybe what's at their vertices are certain gene expression patterns. Right? What's a point here? So if you have, let's say, an archetype is a gene expression profile. An archetype is just a certain partition of, it's just a gene expression profile like you get from a microarray. So it has some biological meaning. So maybe from that you can infer what the tasks are. See what I'm saying? So it's a way to think about cells too. And look at the top 200 promoters that account for 90% of the activity, promoter activity in E. coli. So we're talking about the top 200 promoters. So it's a 200-dimensional space. This 200D data falls on a line, a one-dimensional object. And it looks like this. This is one archetype. This is the other archetype. And here is the gene expression programs. This archetype here, we look at what the genes are, high here. This is growth. What's high here? Ribosomes, transcription, things like that. We look at what's high in this archetype is survival, stress response. We put cells into fresh test tube. They start here. As they finish their food, they go here. You take two different bacterial species, and you put them in the same condition, and you find that two different bacterial species can interpret the same condition differently. One says, in this condition, what I'm going to do in the next time unit is grow. And the other one says, I'm going to grow, but I'm also going to allocate some for survival, because I grew up in a tough neighborhood, and I'm suspicious of what's going to happen next. So this is an interpretation problem. E. coli needs to read the environment. It reads the environment and decides basically on one coordinate, one number. This number, theta, goes between 0 and 1. If 0, I'm here. If 1, I'm here. And all I need to do, basically, for those top 200 genes, is decide on one number, where I am on this line. I want to tell you biologically how E. coli does this. How does it convert its information to a one-dimensional response? It's almost built to do that. You can say it's biochemistry. But did I explain myself here? Okay. I have this Darwin Finch observing me all the time, judging maybe what is it thinking. I've been around for a million years, and now you're reducing me to one dimension.
Are the data points single cells here?
Data points here in this particular experiment are average over a microwell, 96 well. If you look at single cells, it's very interesting. I'll try to get to it. I'll just say that single cells, if you have the population here, single cells form a cloud. And from other data, not this, what we believe happens is the cloud isn't random, it's oriented along the line. So noise gets shaped by the transcription networks in the networks of the cell to go along the line, which is nice because then means you're ready for potential niches. Because off the line, you're always suboptimal.
All these complicated inputs and outputs, they collapse into some sort of simple constant parameter.
Yeah, they do. They collapse on a single parameter, and I want to tell you what that parameter is, so you can have a satisfying sense of closure about molecular biology. So here's how transcription in E. coli works. You know most of this already. So what I want to say is this one coordinate, theta, is the ratio of two sigma factors. I'll explain in a second. Sigma 70 and sigma s that compete over RNA polymerase. So let me explain this jargon. This is the promoter of a gene. In order to make RNA, this big protein complex called RNA polymerase needs to binds and makes the RNA. And when we talk about transcription factors, their job is just to increase or decrease the probability of this binding and initiation. That's how transcription factors work. So RNA polymerase binds to make, it makes the RNA. Now, in order to bind the DNA, it has the sigma unit, subunit of RNA polymerase, is the one that actually binds the DNA. And there's two sigma factors, sigma 70 and sigma S, two different sigma factors that compete over the same RNA polymerase. They compete. And in the DNA, each gene has its sigma 70 binding site and its sigma S binding site. So when RNA polymerase is bound to sigma 70, it recognizes this site. And when it's bound to sigma S, it recognizes this site. So these are two different strings of letters. Each gene carries a lot of information. It's the relative strength of the sigma 70 and sigma S binding site has to be tuned by evolution. Okay? But all E. coli needs to do is you have the environment. All these signals impinge on a signaling network that determines basically the stability and the production of sigma 70 and sigma S. So this theta is sigma 70 divided by sigma 70 plus sigma S. When I write sigma 70, I mean sigma 70 bound to RNA polymerase. So what fraction of RNA polymerase? Fraction of RNA polymerase bound by sigma 70 versus sigma S. This is, of course, a number between 0 and 1. So growth genes, this gene, for instance, this ribosomal gene, has a strong sigma 70 and weak sigma S sites. And this stress gene here is the opposite, has strong sigma S site. So if I have a lot of sigma 70, the ribosomal gene is expressed highly. As E. coli increases the ratio between sigma S and sigma 70, more of these kind of genes are expressed. So the coordinates of the archetypes, each gene, each promoter, encodes the archetype coordinates. So what do you mean archetype? If it was only sigma 70, this would be the expression of the gene here. If it was only sigma S, its expression will be here. So that's determined by these sites. And what it actually does is determined by the ratio between sigma 70 and sigma S, which is on the line between these two. The stress gene has high sigma S binding and low sigma 70 binding. So each gene, evolution can tune the promoters to tell it what would happen if it was only sigma 70 or only sigma S. And then in real life, there's a combination of them. So E. coli has to be very careful to determine the right ratio between sigma 70 and sigma S. And it does this in amazing ways. There's regulation on the production of those proteins, the degradation of those proteins. There's anti-sigma factors that stop them from working. They get phosphorylated. They get massive signaling. And it's different from E. coli to E. coli. It's different from bacteria to bacteria, like I told you, because bacteria can interpret the same environment differently. That has to do with their signaling system. But once they determine the ratio of sigma 70 and sigma S, that's enough to specify the expression of all those genes, and you get points on the line. So it's like this RNA polymerase was built, basically, you can say, to compute a one-dimensional Pareto front. Let's take a nice deep sigh of relief. So, this is for us right now is the best worked biological example of how you can have a protein system that gives you a line. It's still open. How you can have a protein system that gives you a triangle, a tetrahedron. I mean, it's great research problems.
These examples now are in the theorem. Basically, you assume that the strength is always growing. But when you take some problem like with 2,000 measurements, most of those axi are going to be random or close to random. And in that case, basically they're not monotonically growing.
Right.
How do you recognize this?
So this question is about the reality of high-dimensional data, where a lot of dimensions, a lot of things we measure are noisy, or where signal-to-noise ratio is not so good. And here there's a whole skill set about dealing with data. I think next year's course in systems biology will have modules about that. But you can certainly do some things. For example, take the 200 genes which are strongest. That means you're probably far away from the measurement noise. So that's what we did here, and that's very good to start with. Right? So you throw away the genes that are low as the first approximation. And then there's also other things you can do, but they're too advanced for this course. In other words, I don't know, but they're too advanced for this course. But no, there's different things you can do about to amplify noise from data and to tell them apart. But, yeah. Mm-hmm.
Could it be that like high dimensionality constrains each other so that in fact the real space is not so big after all?
That high dimensions constrain each other?
So that you end up with
Yeah.
space.
It's not an easy constraint like the sum of the sum of all these fractions have to equal 1, but that constraint only removes one dimension.
But finite is a resource.
I think the reason for this line is actually natural selection, and you can think, how can you do experiments? Like for example, if you take an E. coli and engineer it to be off the line, over evolutionary time, you expect from this theorem, you will go to the line, for example. So those kind of experiments. But what you need to do is actually perturb 200 gene expressions, or maybe fewer. And so there's certainly cases where I'm thinking of one last little piece of information for you. Or it's not information. It's just a nice way to finish, I think, because if you've ever wondered about the difference between your hands and your feet, I know most of you have spent a long time thinking about that. So I was obsessed with bird toes for a while, since Kathy Kavanaugh showed me some data about that. So if you look at these phalanges in the bird foot, these are these parts of and you look at their areas. And you do again these dimensionless ratios, you find different birds fall on the line, different bird species. And here are the ones that have equal-sized segments, and the ones that have long, short, nothing. And here, if you look at these birds, these ones grab the branch, and that's equal-sized segments is ideal for grasping. And these ones are here like ostrich or walkers, and that has to do with biomechanics. And that's the difference between your hand and your feet. So if you look at your hand, it's like equal-sized, and your feet has long and short. So that's cool, right? Now, what I just want to say is that if I just look at the chicken here, this is the average over all chickens measured. Let's say 100 chickens, this is there. What happens if you grow individual eggs, and you look at the individual chickens? This is the species average. And now you look at individual chickens. They form a cloud. Each one of us is different. Each one of us came from two parents, which are genomes which have never been tested, and their combination has never been tested, but most chickens work. And they're different from each other. So you can ask, is this cloud just a cloud like this in morphospace, or is it aligned along the Pareto Front? And Kathy Kavanaugh did that experiment, and it turns out that individual chickens are aligned with the Pareto Front. In other words, if you exaggerate the difference between two individuals, it looks like other species. The noise and the genetic variation in animals is channeled by the developmental process to a particular direction in a morphospace. And that direction makes sure that you don't have phenotypes that go off the line, usually. And that means you're preparing your offspring for potentially succeeding in one of the niches available to the species. The way we look at it, the reason for this polymorphisms or differences in the genomes between chickens is because chickens have a range of niches, and they need to weigh those two tasks, grasping and walking, differently. So they have selection on genetic variation, but moving along the line. And if you have things like a certain kind of additivity along those polymorphisms, you can guarantee the different parents, the offspring aren't an average of the parents, but at least will be on the same front, and therefore be an optimal solution for multitasking in a particular niche that's available to the population. And if things change and the chicken now needs to evolve to a completely new niche, you have plenty of genetic variation along the front in order to purify out the ones in the wrong direction and zoom along the front in the right direction. That's also observed when you do breeding experiments. It gives you a way to quickly go to new niches. And you could say that these animals, birds, or over their tens of millions of years, have learned the range of niches because of the tasks that are available to their kind of animals, and encode that into their developmental program to learn a particular line. So we talked about multitask optimization, Pareto optimality, and how it can lead to, in some cases, a kind of a simplicity, low dimensionality in polygons in biological data. So let's take a nice deep breath and say, give thanks and praise to our hands and feet, the rest of our parts of our body, which are such wonderful machines. Okay, see you guys next week.